Hello, my dear students. Myself, Dr. Pitambar Humane, founder Botany for You, and working as Professor and Head Department of Botany, Dharampeth MP Dev Memorial Science College, Nagpur, Maharashtra, India. I welcome you all in this online platform for the students of Life Sciences Botany for You. Friends, if you are new or not yet subscribed our YouTube channel, then kindly like, share and subscribe our YouTube channel Botany for You. And if you want to associate with us through the Telegram group, then the link is given in the description. Through that, you can join the Telegram group and can take the advantage of the literature assistance as well as career opportunities in life sciences in India and abroad. Friends, in our last lectures on the genetics and molecular biology, we have completed 19 lectures which you can find in the playlist of our YouTube channel. The last lecture was the DNA damage, and today we are going to continue that series of lectures on genetics, and today's topic is DNA repair mechanism. As you know, there are the many factors which are responsible to damage the DNA, but in every organism there is mechanism to restore that damaged DNA, and that is nothing but the DNA repair. So we'll see the DNA repair and we'll revise that DNA damage also. As we have seen this figure in the earlier lecture, that is the DNA damage. The DNA damage is because of the hydrolysis, deamination, alkylation and even the oxidation, which are responsible for the intercalation of the DNA molecule, then formation of the intrastrand cross, then the interstrand cross, even formation of the radicals, then single strand break, double strand break, then apyrimidinic side, that is the removal of the pyrimidine or the apyrimidinic side, then hydrolytic deamination, removal of the amino group, then apurinic side, that is the removal of the purine, then base damage, change in the nitrogen base, then DNA protein cross link on the DNA molecule, then pyrimidine dimers, formation of the dimers in between the thymines, then alkylation, that is the addition of the methyl or the ethyl group to the particular nitrogen bases which are present in the DNA, then 6,4 photoproduct, then bulky adduct and even the loss of the nitrogen bases. So these are the different ways that are found in the form of DNA damage in the different organisms. But after the DNA damage, every organism try to restore that DNA damage so that there will not be the mutation or it may not lead to the mutation. So how that damage is restored, that we will see in this lecture, that is the DNA repair. If we see this DNA repair, that is classified into the different types. Number one, that is the DNA proofreading, then excision repair, then double strand break and the direct repair or the photoreactivation. We know if we see the DNA which is made up of the phosphate, then sugar, which are responsible for the formation of the backbone of the DNA molecule, and the presence of nitrogen bases, that is ATGC. Just you can remember that the at Gandhi chowk. Now, if we see this attachment, that the sugar is there and fifth carbon atom, here first carbon atom there is a presence of such type of the nitrogen base, either single ring structure or the double ring structure. And so in this way, here there is a attachment of the different nitrogen bases or the 4 different nitrogen bases, and as we have seen in the Watson and Crick model, the DNA is double helical structure in which there are the complementary base pairing in between the adenine and the thymine and the guanine and the cytosine by the 2 hydrogen bonds and 3 hydrogen bonds respectively. If there is change in any of the nitrogen bases by means of the hydrolysis or the alkylation or the dimer formation, it leads to the change in this base pairing or the distortion of the DNA strands. And hence, it is important to restore if the changes are there or the damage is there to the DNA molecule. And hence, even it is carried out during the replication, as we know this process of the replication. Replication is nothing but the process of the formation of the DNA molecule from the existing DNA. And hence, during the synthesis of the DNA molecule from the DNA, here the different DNA polymerases are involved in the proofreading. As in the DNA replication, one of the strand is template and the other strand is synthesized on it from 5' to 3' direction. During this, when the nucleotides are added to the template, again they are proofreaded that they are complementary or not. So, if they are mismatched or the correct nitrogen base is not there, then it is removed and the correct nitrogen base or the nucleotide is replaced by it. So, such type of the proofreading is carried out during the DNA replication or the DNA duplication. In some cases, there is excision repair. Excision means to cut and repel or to remove. There is excision of the base, excision of the nucleotide, and even the mismatch pairs or the bases that are also removed. Then even there are the single strand and the double strand break, and that are also repaired during the DNA replication. And the most common method, that is the direct repair, means when there is a ultraviolet damage to the DNA molecule, it is restored by the visible light, and that is known as the photoreactivation. As a part of the syllabus in the UG classes in the university, we will see the excision repair and the photoreactivation, that is the direct repair, in this lecture. Now, first one, that is the base excision repair. Now, if you see the base excision repair, excision that we see, it is nothing but the removal. And what is removed from it? That is the base, and this base is nothing but the nitrogen base. If there is one of the damaged base, either say for example by the methylation, because if there is a methylation of the guanine which commonly pair with the cytosine with the 3 bonds, and in such cases if it is methylated, at that time instead of pairing with the cytosine, it bind with the thymine, and hence later on the GC pair will be replaced by it. So, to avoid such mutations in the DNA molecule, there is necessity to remove such a type of the damaged nitrogen bases by removing it from the DNA molecule. And such a type of the method in which one of the base is removed which is damaged by one or other means, that is known as the base excision repair. In this process, first of all, the DNA glycosylase find out or recognize the damaged nitrogen base in the DNA molecule, and it cut it from the glycosidic bond, and the damaged DNA is expelled out from the DNA molecule. And in this way, here there will be absence of the nitrogen base, and this is known as the AP site, a purinic site or the pyrimidinic site, means without purine or the pyrimidine on the basis of the type of the nitrogen base which was damaged. Now at this time, here the AP endonuclease act to cut the backbone of the DNA molecule, so that there will be the formation of the strand which is cut at one of the end, and later on from that cut the DNA polymerase is involved for the synthesis of the correct nitrogen bases. Up to 7 to 8 or up to the 12 nitrogen bases are later on added continuously within a correct sequence, and after synthesis of the particular strand on the template, here by means of the activity of the DNA ligase, the gap is sealed. See here, there was a gap, and this gap is later on sealed here with the help of DNA ligase, and in this way this damaged nitrogen base is later on removed, and the normal condition of the DNA molecule is restored. In second one, that is the nucleotide excision repair. As we have seen earlier, in the base excision repair, there is an removal of the methylated or the damaged nitrogen base, but here if there is a presence of a thymine dimer, in such cases the nucleotides are removed. And in this process, first of all, if it is damaged DNA by means of the ultraviolet light, and the ultraviolet light is responsible for the introduction of the thymine dimer in the DNA molecule. So first of all, it is recognized by the enzyme, and when it is recognized, at that time the different multiple enzymes bind to it, and later on there is formation of the incision to the damaged part of the DNA molecule, and here there is formation of two nicks, that is 3' nick, and second one is a 5' nick, and because of this, here there is a distortion of the one of the damaged strand or the containing thymine dimer. Now here the exposed nitrogen bases which are formed after the nick at the two different ends of the DNA molecule or the DNA strand, here there is synthesis of the new DNA strand from 5' to 3' direction. Up to 24 to 32 nitrogen bases or the nucleotides are added in this section, and again here it is continuously synthesized, and then with the help of DNA ligase, here the gap is sealed, and in this way the damaged part of the DNA strand is again restored by removing the certain nucleotides from the DNA molecule, and hence it is known as the nucleotide excision pair. In the next type, that is the photoreactivation. Earlier I told you that the ultraviolet light is responsible to introduce the thymine dimer in the DNA molecule, and when the thymine dimer is established, first of all here the photolase enzyme recognize that thymine dimer which is formed during the exposure by ultraviolet light. And this photolase again at the another enzyme, that is the DNA chromophore, and this DNA chromophore is responsible for absorption of the visible light. If you see this process of the photoreactivation, here after the ultraviolet radiation there is need of the visible light within 1 hour. So directly it can be restored in a normal condition, and there is a mechanism in every organism for the photoreactivation. Now at this time, when the enzyme DNA photolase and the DNA chromophore added to the thymine dimer of the DNA molecule stand, then that particular nitrogen bases which are forming the bond in between them, that is the two thymine molecules or the thymine bases, here this bond is broken, and hence here there will be formation of the separate nitrogen bases or the thymine molecule. Now again, after breaking this bond with the help of visible light, again the repair of the DNA and release the enzyme. So here, if this bond is broken down, automatically it will store the normal condition of the DNA molecule. As there was a formation of the dimer and distortion of the DNA molecule, as it was unable to form the hydrogen bonding to the complementary nitrogen bases. Now as this bond is broken down, it becomes normal and the damaged DNA is restored. And in this way, even if the DNA molecules are damaged by the ultraviolet light, then it can be restored by the visible light in the organisms. And such a type of the process is known as the photoreactivation. Friends, this was a simple explanation with respect to the DNA repair mechanism for the huge classes, and I am very much hopeful and confident that you must understand the meaning of DNA repair mechanism. If you like this lecture and if it is useful to you, then kindly like, share and subscribe our YouTube channel Botany for You, and also press bell icon for the alerts of every lecture to be uploaded on different life sciences titles. In next class, we will see the types of RNAs. So thank you very much for watching this lecture completely.